Okay, welcome to this explainer. Today, we are going to unravel a massive biological puzzle, the one that Gregor Mendel actually solved way back in the mid-19th century. Have you ever wondered exactly how traits are passed down from generation to generation? I mean, why do we look the way we do? Well, today, we're digging into the specific, hidden mechanisms that dictate all of that. And it all starts with something as simple as a garden pea. To make this complex topic super clear, we've broken our journey down into 5 quick steps. We'll cover the core mystery of inheritance, Mendel's peas, the monohybrid cross, how to decode a Punnett square, and finally, Mendel's first 2 laws.
All right, let's jump right into part 1, the mystery of inheritance.
So here is the fundamental mystery we are trying to solve today. How is it possible for a specific physical trait to completely disappear in one generation, only to magically reappear in the next? Think about it. Imagine a kid having the exact same distinct nose as their grandfather, even though neither of their parents has it. How does that biological information secretly travel through time? Before we can crack this puzzle, we need to establish a quick foundation. First up, inheritance. This is the absolute basis of heredity. It's the actual process of passing characters from a parent to their progeny. Then we have variation, which simply explains the degree to which those progeny differ from their parents. These 2 concepts are the bedrock of everything we're talking about today. Now, what's absolutely wild is that humans had been selectively breeding plants and animals for thousands of years. As far back as 8,000 to 1,000 BC, humanity already knew for a fact that sexual reproduction caused this variation. We knew how to exploit it to get the traits we wanted. But the actual biological code, the physical mechanism making it all happen behind the scenes, remained a total mystery. That is, until one man stepped up.
Moving right along to section 2, Mendel and his peas.
Gregor Mendel didn't just take a wild guess. He literally spent 7 years, from 1856 to 1863, conducting incredibly rigorous hybridization experiments. And he didn't just pick peas at random either. He chose them because they offered massive, highly reliable sample sizes and super stable traits. By utilizing 14 true-breeding pea lines, meaning these were plants that had undergone continuous self-pollination for several generations, he could easily master artificial, controlled cross-pollination. The sheer scale and dedication of his work was just astonishing. Mendel brilliantly isolated 7 specific pairs of contrasting traits, setting up this perfect binary framework. Stem height, it was either tall or dwarf. Seed shape, round or wrinkled. There was no in-between whatsoever. This simple binary setup was genius because it allowed him to clear away all the genetic noise and track exactly how a single character was passed down. And here is the real game changer. Mendel approached biology like a mathematician. Actually, this was the very first time anyone had applied statistical analysis and mathematical logic to problems in biology. By meticulously tracking the exact numbers of these traits across successive generations, he gave his data immense credibility. It proved his results weren't just random ideas, but actual universal rules. Which perfectly sets us up for section 3, the monohybrid cross. To really test this mathematical approach, Mendel initiated what we call a monohybrid cross, which is just a fancy way of saying a cross to study the inheritance of one single gene. He took a true-breeding tall plant and forced it to reproduce with a true-breeding dwarf plant. Then, he collected the resulting seeds, planted them, and waited to see what would happen to this offspring, which is known as the first filial or F1 generation. The results were completely shocking. The dwarf trait literally vanished. 100% of the F1 generation plants were tall, perfectly matching just one of the parents. And crucially, there was absolutely no blending. You might expect a medium-sized plant, right? Nope. It seemed as though the dwarf trait had just been erased from existence. But Mendel didn't give up. Instead, he essentially asked the plants to clone themselves. He let those tall F1 plants self-pollinate to create a second filial or F2 generation. He needed to know, was that missing dwarf trait actually gone forever, or was it just secretly hiding inside these tall plants? And guess what? The dwarf trait made a dramatic return in the F2 generation, but it didn't just pop up randomly. It appeared perfectly mathematically, in a ratio of 3 tall plants to every 1 dwarf plant. Exactly 1/4 of the plants expressed the dwarf trait, and 3/4 were tall. And again, 0 blending. So how did that math magically work out? Let's figure it out in section 4, decoding the Punnett square.
First things first, we need a little vocab to separate the outside from the inside. Based on what he saw, Mendel proposed that something stable was being passed down through the gametes. He called them factors, what we call genes today. Now, the phenotype is what you physically see, like a tall or dwarf plant. But the genotype, that's the hidden genetic code driving it, usually represented by letters like a capital T for tall and a lowercase t for dwarf.
Next up, Mendel realized that these genes, which code for contrasting traits, are called alleles, and they always occur in pairs. Plants can carry identical pairs of these genetic factors, which we call homozygous, think 2 capital T's or 2 lowercase t's, or they can carry a mixed, dissimilar pair, which we call heterozygous, like 1 capital T and 1 lowercase t. Okay, this is the real aha moment that explains the F1 mystery. When you have a dissimilar pair of factors, one is always going to dominate the other. The tall trait is a dominant factor, so it simply masks the presence of the hidden recessive dwarf factor. That means a heterozygous plant with 1 capital T and 1 lowercase t will still physically look tall, because that dominant capital T is completely running the show.
A bit later on, Reginald C. Punnett gave us this absolutely brilliant visual tool to calculate these exact probabilities, the Punnett square. When those heterozygous parents from the F1 generation create gametes, pollen and eggs, they randomly pass on either a capital T or a lowercase t. When you combine them mathematically, box 1 gives you TT, which is tall. Boxes 2 and 3 are TT, also tall. But that fourth box, that's TT, our recessive dwarf plant. And boom, that is exactly, mathematically, where our 3-to-1 physical ratio comes from.
Finally, let's tie this all together in Section 5, Mendel's first 2 laws. From all these incredible observations, Mendel formulated the law of dominance. It establishes 3 huge rules. 1, characters are controlled by discrete units he called factors. 2, these factors always occur in pairs. And 3, in a dissimilar pair, one strong dominant factor will completely dictate the physical outcome, masking the recessive one. This perfectly explains why every single F1 generation plant was tall. His second massive breakthrough was the law of segregation. Since we know alleles don't blend and the hidden traits recover in the F2 generation, this law guarantees that during gamete formation, the parent's pair of alleles randomly segregate or separate, so a gamete only receives 1 factor. It's a perfect 50-50 chance for either allele, which keeps the inheritance cycle totally balanced and lets those hidden recessive traits safely travel across generations. So we've officially mastered the inheritance of a single gene and unlocked the monohybrid cross. But I want to leave you with this provocative thought. We know how 1 pair of alleles behaves, but how chaotic does the math get when traits like seed color and seed shape are inherited simultaneously? What actually happens when you cross plants that differ in 2 completely different traits at the exact same time? Well, that dihybrid cross is a biological puzzle for another day. Thanks for joining me on this explainer, and keep questioning the science around you.